Good morning. It's a bit strong. Good morning, everyone. I am Damien Beigel. My PhD was a few years ago. The sound is a bit strange, no? Yeah, anyway. And I'm here to give you a talk. First, I would like to thank all the volunteers. I think we can thank all to you. And it's, you know, every people is important, so it's good things to remind. Just to remind, we saw yesterday you enjoyed a lot the hands-on lab session. It was really great for us. But it's important that we respect the time. So when it will be finished, it will be finished. Even if you want to continue, I know it's a bit frustrating, but you know, we have to maintain the good schedule. So we'll finish at 5, I think, maximum today, right? Okay, so today I would like to talk. Yesterday I tried to explain you why it could be interesting to think about programmability in soft matter. Can you? I don't know. It's a bit strong here. And before that, I would like to give you a brief recap so that we can start from the same background. So I remember I showed you a type of soft matter that are living systems which have really amazing properties, which are thus interesting to study but complex to understand. At the same time, from more engineering point of view, it could be really nice if we can build such kind of properties. This is what I show you, and at the same time, we living systems are soft matter, and for two main reasons. Is that first because we are made of soft matter components such as polymers, colloids, interfaces, and at the same time most of biology works at thermal energy levels. So the energy are typically comparable to thermal Brownian motion. So we are soft matter, and at the same time, and also I try to show you that even if soft matter is very diverse and broad and familiar and interesting, what is quite interesting is that we can find some unity in this diversity, and we can say that it's basically everything working around the thermal energy KT. So it is possible to have some laws even despite of this dispersity, and with that, it's nice because maybe we can approach what I call lifelike properties, which are all these dynamic self-organization, sensing reconfigurability, adaptability, or motions that living system can have, and then that we can observe and do with synthetic system. At the same time, I think it's quite nice to work with precisely synthetic soft matter systems, because first they are well controlled, so you know what you put inside, so you have a possibility to study something. You can control the parameter, you vary, and this is not something you can do with life, right? You cannot change the parameters of life if you want to study it. And at the same time, if you think about building objects, it's quite nice to have something synthetic, because then you can adapt to your needs. So you have custom design possibility. So, as I say, we have these two motivations to understand and to build and to make. And I try to show you that if we look at how nature is working, which is based on this huge collection of functions that can ensure by proteins, if we look at all these functions, they are really great and diverse, but at the same time they are very complex. And what is quite interesting is that in living system, how life is working is to encode all this diversity in the same molecule, in this very same molecule. It's the DNA. And DNA is very simple if I compare to proteins. So DNA is a molecular code of life, and it's shared in the same molecule in all living organisms, and it's the support of the code that we are talking about. And at the same time, it's a very simple molecule. So life is working like that. And I know I ask you to be careful and give me your attention, because now, okay, I made this recap, but now we'll try to go forward and see how we can do with that. So in nature, you have this interesting separation between a program and the functions. And program is in a very simple molecule that you find everywhere. It's highly conserved. And functions are in this huge diversity of proteins, and they are this complex molecule. So you have this separation. And quite interestingly enough, all these important components, the code that the DNA and the functions are proteins, they are soft materials. They are soft materials because they are polyelectrolytes, and I explained yesterday, because they are polyelectrolytes, it's very important for the code because this allows the DNA packaging, so it allows information storage. If DNA would not be a polyelectrolyte, we could not condense it in each of our nucleus. We have 2 meters of DNA in each cell. So imagine what you have to pack, and it's a charged molecule. So it's important for information storage. And if you look at proteins, it's important that they are polyelectrolyte behavior because it helps them to interact. And also soft interactions. Both DNA and proteins work through soft interactions. It's very important for the double helix maintenance, but also the possibility that life has to separate the DNA and replicate it. And this is extremely important for information transfer from one person to its progeny. And in terms of protein, of course, most of their functions are inside their dynamic conformation. So it's very important that they are built with soft interaction and not just with covalent bonds. Okay, so if the structure was held by covalent bonds, it could not be so reconfigurable. Okay, so what I would like to talk today and tomorrow is that life has very interesting property. We would like to understand and we would like to make these life-like properties. And we know that soft matter is perfectly suited for that because life is part of soft matter and soft matter is in life. So the idea is to make synthetic soft materials, so materials that we can design, materials we can control, but instead of doing formulation, like what people would do usually in cosmetics or in polymer science, they will do formulation. The idea here is to put a code inside, and our code will be DNA. And with this code, we'll try to program either structure, information, either functions, so that we can get all these kind of properties. So this is a bit different concept that usually when you prepare something, a formulation, you will put many things somewhere to get your property, and you will repeat your experiment until you get your good property. Here the idea is to try to be a bit like how life is working, that I have my system, and inside I will put the information, and all the information will be here, and the system itself will be able to build its functions. And what I would like to code is both structure and functions, and with that I would like to approach all these lifelike properties that I have introduced yesterday, like reconfigurability, evolution, environment sensing, and maybe even motion. Okay, could we make something move with the DNA inside? Could we make, for instance, let's take some Indian soft matter. You have a lot of soft matter in food. You take your favorite food and you want to make it move, okay? And you don't want to make it move mechanically, but you would like to make it move by itself. So you would like to put a DNA inside so that this could move, okay? This is what we will try to do today. It's quite challenging, but I will show that maybe there is some ideas we can try. So I would like to ask you now 2 questions. Why should we choose DNA as the code or as the molecular program? And the second is how to make it. How to make this program. So I'd like to ask you, why should we choose DNA and not something else? Okay, we can say it's because life is working like that, but me and Arnaud in a building synthetic system. So why do you think we should use DNA as our code? The code as my molecular program. You know, you understand, I want to have something in which I want to put information. it should be a molecule, and why DNA is interesting. So you say DNA is more stable?
With respect to RNAs and others, a genetic material.
Yeah, but why do you restrict to RNA and enzymes? Why not taking a polymer, polystyrene, for example? It's very easy to synthesize, maybe easier than DNA. Why not using synthetic polymer? There was somebody working with polymer, right? I think so, few people, yes? Like block copolymer. Why not using block copolymer? Why using DNA?
Because we can produce a desired protein to work with if we just use coding with DNA.
Yes, so, but if you say we can somehow encode proteins, why make this possible? Because here, I would like to build a code, so I need information, right? So how can we have information in a polymer? Yes, so...
Yeah, we can like design the DNA as per all the nucleotides, and then maybe we can use a complementary one to have interactions between them.
Yes, so DNA has this possibility that we can have a sequence of nucleotide. There is only 4 that we sequence. It's true, and this is something very nice, but I don't know if there are good polymer chemists here, but you may know that it's possible to build polymer, block copolymer, right? So why is DNA more interesting than a block? So block copolymer, you could put some information inside, right? So and even people are doing it. So what could be interesting in DNA? What could we have more with DNA compared to a regular block copolymer or statistic?
They can be cross-linked, maybe. Cross-linking possibility. Proteins.
Proteins. So let's stay at the molecule, at the code level, at the program. We maybe speak about.
They are diverse and can be make combinations.
Yes.
That's it.
But still, you know, if we know well the chemical synthesis, maybe we can synthesize very well-defined polymers and put information. You know, there are people who are storing information in DNA, of course, but also in some synthetic polymers. So, yes, any other ideas from this side? Because I don't know why everyone is... Yes? The structure is very stable of DNA? So, more stable than what?
Let's say compared to other genetic, other polymers like RNA or something.
Yes, you compare to RNA, but for example, if I take this telephone, I think I can drop it. So you know, this is very strong. This is very stable. Would you like to make it out of DNA? Do you think it will be as stable as this? It's true, DNA is a very stable molecule. We can find DNA from dinosaurs, you know, so it's stable. That's why some people consider to put information in DNA because it's very, let's say it's true that it's quite stable molecule, but compared to many other synthetic polymers, it's maybe not the advantage. I agree that compared to proteins or compared to RNA, it's much more stable. That's right. But compared to other polymers, maybe not so much. Okay, so I think we have some ideas a bit. And I think the two key advantages of DNA is because here, it's for me, is two things, the accessibility and the programmability. Programmability, we mentioned quite by several people here. It's true that we can make a sequence of nucleotide, and this can be information. And there's something also very important, is the accessibility. And this accessibility is possible because you are young enough, and even me, to have witnessed a technology called twist in the past decades, which is DNA synthesis. And in fact, and this is something that is very interesting if I compare to a synthetic polymer, is that maybe there are a few people in the world that can build very amazing polymers in a lab somewhere, but this is something very specific and that require high knowledge, high technicity, and maybe only a few labs can do that. But DNA, everyone can get any DNA he or she wants, and this accessibility is something very important besides programmability. So accessibility is because now we are lucky that there is technology available, and this technology is DNA synthesis, and we can synthesize DNA at any sequence we want by simply using synthesizers, so it's our machines, and you just provide nucleotides. It's based on solid state photoharmonic chemistry. So basically, you have a solid support, you put one nucleotide, you rinse, put another one, you rinse, put another one, and like that you can build any sequence you want. So you can really choose the sequence you want. And this is for mainly oligonucleotides, but you can also get long DNA by using assembly and combination. And for instance, oh, I didn't expect this animation. For instance, it's called Gibson assembly, where you can combine and build very long DNA. You know that people have been able to synthesize whole genomes of organisms. Okay, they could take, for instance, some bacteria, remove all the DNA, synthesized everything, put inside, and make an object alive. So you see that we start to be at a level that it's possible to build DNA, enough DNA to make an organism alive. Okay, so it's quite amazing in terms of technology. And it's accessible in the sense that you can synthesize by yourself if you have a synthesizer, but you can also just purchase. So you don't even need to have your machine in your lab. You can just order to any company your DNA. And this is actually inexpensive. Oligonucleotide, it will cost $1 to $2 per oligo. And if you want to ask to synthesize some genes, it's also reasonable. It's like something a few cents per base. So if you take a gene that is like 1,000 base, it will cost you maybe something like hundreds of dollars. And once you get it, you can replicate. You can replicate as much as you want. You just give to bacteria and you replicate it. So you don't need a lot of materials you can produce in large amounts. So we have this very interesting accessibility of DNA. It's very important, of synthetic DNA. And at the same time, you have full programmability. Nowadays, you can basically get any sequence of DNA and any length you want. You just have to have a good idea, and you can get it. And you know that people, for example, they demonstrate that it's so powerful and so accessible that there are people who could order DNA to synthesize a virus, okay? A virus, for example, for smallpox, so a disease that nobody vaccine anymore. So you see that it's really powerful. So as anything that is powerful, of course, there are ethical issues, we have to be careful, but at the same time, it shows how powerful it is, this method. You can basically just decide what sequence you want, and then you can order and you can get it in your lab, okay? And it takes just 1 week or so. And at the same time, you have also what is very nice, besides the sequence, you can add functionality. So you can ask an oligo the sequence you want, and you can ask to add, for example, an acid carboxylic, or an amino group, or click chemistry group, or a biotin so that you can interact. So you can add functionality in your DNA. So you have this full programmability, and you have this complete accessibility. Okay, so this is not something that you can really get. You are just limited basically by your imagination and your ideas. So now that we have this DNA, that we can have complete access and whatever the sequence you want, the idea is with this we can program functions or we can program structures. And because this is now established, this led to the development of two completely new and independent fields. On one side, in synthetic biology. So you can get any DNA you want, and then you can start to reprogram cells. You can start to control how the life will do on a synthetic level. And on the right side, you can program structures, and this gives rise to structural DNA nanotechnology. We can build objects out of DNA that are really amazing, and I will show a bit of both today, this, and tomorrow I will show you more on this side. As an example of synthetic biology, you can take DNA and you can reprogram cells. So for example, you can take a cell, add synthetic DNA inside, and you can ask these cells to behave in an interesting way. For example, if a cell starts to have a cancerous behavior, you can make a program that the cells will decide to kill itself. Okay? So you see it's very different approach. Like usually when you want to kill cancer, you put drugs, you put things that will kill cells. And of course, it's a very brute approach. First, you need to find the good cells, and you need to kill them efficiently. At the same time, you don't want to kill the other cells. Something very difficult, and cancer is very difficult for that, is because you have to kill the cells of a given person. So with synthetic biology, you can put program, for example, to tell cells, okay, if you become cancerous, just kill yourself, and you just use the biology to do that. So it's very interesting as an approach. And because we are lucky enough in this room, because we are all soft matter scientists, and as I explained, biology works at soft matter level. Katie, DNA is a polymer, is a soft material. Proteins are polymers, are soft material. So we can use soft matter to do that. And it's not so usual. If you go to this field, if you look on internet synthetic biology, you will almost never see people speaking about soft matter. So this is soft matter. And if you go to DNA nanotechnology, you look on internet, you look at this great community, you will see that very few persons speak about soft matter. But they are working with soft material. So there are really, I think, a lot of opportunity in these two aspects of synthetic biology and structural DNA technology if you come from soft matter. There are really a lot of things to do. So today I will speak about this part, programming functions, starting from the synthetic DNA, in the framework a bit of synthetic biology and in the context of soft matter. So I will now go to a more detailed talk about this specific topic. Okay, so now I will try to speak more specifically how we can program mimic biological, non-biological functions at this interface between soft matter and synthetic biology. So, as I said, as I remind briefly at the beginning, in our everyday life, in industry, there are a lot of soft materials that are very interesting for their property, and have a lot of applications. But many times, when people build soft matter, they do it by formulation. So they will try to add everything in good proportion. Like when you cook, you have to, when you deal, for example, with spices, you need to know to put all of them in a just good proportion. You need to find a balance so that you have a very good dish at the end. Okay, this is just for taste, but you have to do this for viscosity, you have to do this for mechanical properties, you have to do this for chemical properties, so you have to do this adjustment. And this can be great, but it's very system specific. It means that if you change your system, then you have to do this again and again and again. Each time you have to repeat this approach. And also, most of the time, you can get very nice object at the end. You can get, for example, a wonderful dosa. Okay, you get a wonderful dosa. But then this dosa is a dosa, and you cannot transform a dosa in an idli, for example. It's done, right? So we can have you have to do this very complex adjustment, and sometimes you have something that is quite fixed and dedicated. If we could program, if we could have something inside your material, like a DNA, when you will put the property, you will say, in my DNA, I want a crispy dosa, I want a dosa that has a bit of ghee taste, but not too much and so on. And that could be great. And maybe you can put another information that this could also become a very soft idli at some time and so on. This could be nice because then this could be universal. You don't need to care about the specificity of your material, you just put a code inside. And maybe we could have configurability, maybe we could have system that can transform a bit like a living system can do. So basically, the idea is to take a soft material and instead of making it from ex situ approach, I would like to put a genome inside and control all the properties of this object. So let's try to do this, to genetically encode synthetic soft matter to control both properties and functions. So we say nature is working like that. Let's try to do the thing. First question is, what will be the program? And we already answer the program that will be used in synthetic DNA, because we have programmability and accessibility. So first question is, what will be the functions? What kind of functions we can do from the DNA? How we can convert this information we have in synthetic DNA into functions? What will be the molecules that will ensure the functions? From my synthetic DNA? It's great to have a program, but if I cannot convert this program into a function, it's a bit useless. So it's very simple. What will be the functions? What kind of molecule I can get from DNA? What kind of molecule? Right? Peptides, yes. Peptides, yeah, very good. I can put information even more general and even more a bit complex than peptides. Yeah, but look, in nature, you have your program that is DNA, and this program is transformed into function, proteins which answer a function. So we are synthetic, we are on the right side, so our program is synthetic DNA. So it's not a DNA I will take from a set, it's a DNA that I will synthesize with a synthesizer. So what will be my functions that I can get from DNA? It can be peptide, but it can be even a bit more elaborated than a peptide. Yeah, but DNA will be still more information. So I speak about functions. Proteins, yes. Why are you so shy? Suddenly, you say DNA is super easy, but proteins, yeah, this will be our functions. Now we have DNA. So it will be proteins, but the specificity is that this protein we want to be made in situ. We want these proteins to be made in situ, okay, in our synthetic system, from our DNA. We don't want to order a protein, so we can ask, but this is not what we want to do. We don't want to take a protein from somewhere else. We want to have our DNA that produces this protein. Okay? So we want to have DNA, and we want to synthesize these proteins from DNA. So how we will do that? How we will transform our DNA into protein? Can you do that? Can you, if I ask you, if I give you synthetic DNA, can you make for me the protein? Who can do that in this room? Hmm? DNA like this? Yeah, but now we have a specific goal is to make proteins. We want to synthesize protein. Usually this is done by cells. Like us, we are now synthesizing a lot of protein. So they are very kind, the cells, and they are working hard to produce proteins. But now we want to do everything synthetic. Everything is dead, not alive. So
You need enzymes for transcription and translation process.
transcription, translation, without cells, right? We don't want to use living system because we want to be synthetic. So yes, we will do. So cells do protein synthesis, and what we will do is in vitro protein synthesis, and we'll do cell-free protein synthesis. Okay, so we want to produce protein in a totally cell-free manner. So the question is, what do we need to make protein? So we have DNA. And we need to go to protein. So what do we need to, how is it working in cells? So first DNA is made, make some RNA. And then the RNA will be a protein. So yes. Okay. So now we want to do it in the lab, okay, without cells. So we can try to know what do we need. So what do we need to go from DNA to RNA? So we need polymerase. It's what is polymerase? So it's a protein. Here, what do we need? So tRNA will be synthesized. So we'll have some breaks.
Ribosomes.
So ribosomes. We need amino acids. So amino acids, I would say it's more like break. So this, I would say, these are protein. So I will put like, this is protein. So we need bricks, amino acids. There are 20 amino acids, different amino acids. So these, I would say, are different breaks. So we need some breaks. We need the precursor to make the RNA. These are other breaks. So we need some proteins, we need some breaks. And what is the third element that is quite important if we want to make it work? We need a bit of energy, chemical energy. And the energy is what is the biological, what is the chemical, how biology can work at room temperature with ATP. So we need some energy. So, easy? If I want to do this, instead of using a living cell that is very complicated, maybe I can take just all these components in a tube, purified. Okay. And this, and the energy. And here, I can have a cell-free expression medium. So it's quite interesting because life is beautiful and complex, but actually, it's just working through well-defined principles. So if you just took everything needed that is necessary to go from DNA to a protein, you just take these essential components, and with that, maybe we can go from DNA to protein. And we can do this without anything alive, without any cell. And actually, this is nice because it actually exists, and it's available. And it's called cell-free expression. And cell-free expression is quite cool because it makes synthetic biology very easy and possible. So it's actually very easy to do what usually a cell is doing, is that you take a cell-free expression medium, and what is very interesting is that you can do this, but this is complicated, but you can just buy. It's commercially available. So there are people who have purified everything and put just in a tube. So you can buy this, you can do in any laboratory, you can just buy this, you put inside the DNA that will code for your protein of your choice, you put RNA-free water just to avoid to degrade the RNA that will be synthesized, you incubate at 37 degrees, and then in a few hours you will get your protein. So the only things you have as constraints is that usually the expression system we use are derived from bacteria, because bacteria you can produce in very large amounts. So you can produce bacteria and extract all these components. So bacteria, you can take just a huge fermenter, you can produce large amount of bacteria. So what you need in your DNA, you take a plasmid, so you will put, here will be the sequence of your choice. Okay, so this you order, okay? And what you need is just T7 promoter, because usually the cell-free expression medium are derived from bacteria. So usually they work with a T7 promoter. So here, this will be recognized by the RNA polymerase. So it's important. So if you use T7 RNA, it will be able to recognize this, and this guy will know that it has to start reading from here. It doesn't have to read this guy. You need what is called a ribosome binding site. So because it's bacteria, the ribosome has to bind your RNA. Then the ribosome will read this, make the protein, and then you just need the guy to so this is just a sequence of DNA, 80 CG, 80 CG, and 60 CG, and you just order it. Either in synthesize or you can just order your DNA. It's commercially available, and you can order your cell-free expression medium. And now it starts to be very cool because in your lab, on your bench, you just need a very small bench like that. You take your cell-free expression medium, put in a tube, you take your DNA that you order because you just choose your sequence, and then you put this at 37 degrees, and then the protein will be made in your tube. Just this is what cells usually are doing. You can do it in your tube on your bench. Now you have something that's quite interesting because it becomes very simple. You choose the function you like, you want to do. For example, I want a dosa that tastes cinnamon. This is the function I want. You look at the protein that can give you this function. And this is quite easy because you have so many proteins available in the world, okay? So you will pick up the protein that is function. So this is just what you do with your brain, okay? You don't do anything in your lab, no? And then you just need the sequence of this protein. And this, it's nice because you have so many databases, and these databases are open source and publicly available. Okay, so you just, again, this is, I would say, your brain, and this is, you just said, yes, the data that are known, and here you just use database, so basically internet, you go to internet, you enter your protein name, and you will get your protein sequence. And from your protein sequence, then you can just convert to DNA. It's very simple. You just have the codon, and you can just have a few optimization, but these are very simple. That you can just use online software to do that. Now you have your DNA sequence, and you just order. You send your sequence to any providers, and in 1 week, you will get the sequence in your lab. Okay? So that's very nice, and then you can just do this experiment. This you have already in your lab, you mix with your synthetic DNA, and then in your tube, you will have the taste of your dosa. Okay, and this you can just choose any property you want. So it starts to be quite interesting in terms of possibility you can do. So maybe I would like to ask you, what would you like to do? What kind of functions you would like to do? I'll give you 5 minutes to think about it. And then I will pick up a few of you to tell me what he wants or she wants to do. So just think what could be an interesting functions you can do with some interesting proteins that could be useful or interesting and curious, or I don't know. I'll give you 5 minutes. So I will pick up 4 persons or 5, I don't know. So who is from Kerala? Originally, who is from Kerala by origin? Who is from Kerala? Raise your hand, people from Kerala who were born in Kerala. You can be proud, it's a great region, right? No problem. So I will pick up 1 people from Kerala, right? Huh? You are from Kerala? Okay, come. So I will. Is there anyone from Telangana? Telangana? You are from Telangana region? No? Nobody? Let's say Andhra Pradesh? Andhra Pradesh? No? Nobody? Tamil Nadu? Ah, South India, yes. Tamil Nadu? You, okay, you. Then Gujarat? People from Gujarat? No? Karnataka? You? Okay, so you will be the team Karnataka. Okay, so third person. And maybe last one, Madhya Pradesh? No? Nobody? Bengal? What? Yes, you? Ah, great. Okay, so we have 4 teams, so you will represent your state, so it's very important. So you, what you would like to do? So you represent Kerala, right? Yes? So what you would like to do by using this approach? What kind of function you would like to do with this approach?
Okay, so there's a bacteria called Idoneus accasis which can digest plastics
Okay.
within weeks.
So some enzyme
Yeah.
that can digest plastic, okay.
So I would like to target that protein which produce that
Okay.
particular enzyme.
Very interesting, yes.
so
So
that-
you would like to do this for depollution or pollution removal?
Yeah.
Okay, very interesting. And would you, so what could be the interest of self-free expression? Like what kind of materials you would like to do? Like how you imagine your system, like with droplets, or how you imagine to do your self-free expression? Because okay, you can look at your enzyme sequence,
Yeah.
and then you take the DNA sequence, then you do the self-free expression. So in what kind of, I don't know, materials or what kind of system you would like to do this?
My intention is to produce commercially available chemicals which can digest bacteria. So initially, rather than focusing on biochambers, I would like to form synthetic DNAs which can produce commercially available enzymes. And
okay, so to produce the enzyme, and then you want to do business. You want to sell the enzymes. Okay, it's an objective, no problem. Okay, very interesting. So this was Tim Kerala. Bravo. Yes. So then the next was, who was the next person? Ah, yes. So you are representing?
Tamil Nadu.
Ah, okay. Other people? You are the only one?
I guess I'm the only one from.
Okay, but you have a big big responsibility. Okay, what you would like to do?
So considering this, the procedure, like a reverse engineering kind of thing. So the target function, proteins can perform a variety of functions. So the one I could think of is a similar one, like an enzyme that focuses on, let's say, degrading a particular molecule that could probably lead to like a disease, like help in reducing the effect of an antigen.
Yes. And this you would like to, like here, you would like to produce
an enzyme.
These enzymes, or you're saying to make it inside the body?
Yeah.
Because you can imagine that your DNA you put, I don't know, in some whatever liposomes, and then you give to patient. Maybe you can say, I will express the DNA just where it is needed.
Yes.
For example, these enzymes, that could be very interesting. So enzymes are very interesting because they are catalysts, so they can do a lot of reactions. So enzymes are a very interesting target indeed. I think it's also a very good idea. So well done also.
Thank you.
Yes, you have your colleagues. Yes.
I'm also from Tamil Nadu, and
yes.
I just have one small idea. Like, I'm not sure about this biological system. I don't have that background. But from my master thesis, I worked in a smart material. So what I think, a protein that can change its color, I'm not sure it will happen or something.
Yes.
Like, it can respond to a pH or temperature, like a smart material that can respond for external stimuli. So we can use that for a biosensing and targeting a tumor.
I did some magic. I have a smartphone in my hand. It appeared. Ah, okay. No? This one. You stole my phone. Okay, sorry.
Yeah.
Okay. So if we think about like some kind of response behavior, we have two possible strategy. So one possible strategy is that you put a protein that has this property. Maybe there are in nature some chromophores, some proteins that can change colors, right? Like we have fluorescent proteins, and maybe some can change color. So in this case, you could indeed synthesize this. But you have another possibility, is actually you can also maybe control this reaction. So you can have the protein itself that is responsible, or you can have this expression that is also responsible. So if you trigger this expression by pH, then you can trigger the color, or you can have the protein itself that changes the color. So I think two possibilities. So let's move to the next team. Who was the next team? Who was the next team? Ah, you, right? You are from?
Karnataka.
Okay. So it's the next state, right? It's close.
Yeah.
Yes. So what is your what you would like to do?
The target function.
Yes.
So actually, I don't know anything related to biology.
Not that's okay.
But what I get frustrated is, I have a lot of hair fall.
Yes.
Yeah, like whenever I feel like, like I want to go to medical related research and, bring some, make some proteins that can stop hair fall. That every time I feel that. Yeah.
So sorry, you want to do what exactly?
I didn't understand.
What do you want to do exactly?
Maybe synthesize some proteins that will stop hair fall.
Ah, okay, yeah. I see.
Like,
yes?
Yeah.
Maybe we can think about it. Maybe if there are such proteins, right? Yeah, but that's good. So I'm sorry, but Greg, thank you. What is the team? It was you, actually.
Yeah, I'm from West Bengal, and
West Bengal, yeah.
He actually incepted my first idea, but anyway, I'm gonna say something else. So I was thinking, in terms of farming, we use different types of pesticides and
yes,
all. And also we use some kind of like for growing the plants and stuff. So if we can combine them, make a targeted protein like this, which can also like act as a food as well as the keep the pests away. Maybe, I don't know. It's totally hypothetical.
You think, for example, to produce food?
Yeah, I mean food for the plants, like to grow, as well as it might act as keep away the pests. Both.
Yeah, it's a very good idea. You know that some people I think in the Netherlands have tried to produce by this approach meat,
okay,
synthetic meat, starting from DNA, right? So these things are done, right? So, but it could be also to make some interesting protein. So I will show you, yes? Molecular motors? Yes, molecular motors are interesting types of proteins because they can produce mechanical forces. And this could be nice, for example, maybe in soft matter to put in some gels or in some soft materials so that the soft materials could move, maybe make some kind of robot. Yes, or actin filaments. So it's interesting because of course, enzymes are very nice because we have direct application, but this could be very nice, for example, to make, maybe program some, and then what could be nice is that you can put in the DNA what you want. Okay, so I will show you a few examples, and I will focus on things that are a bit related to soft matter. And we will see there are other kinds of proteins that are quite interesting, actually. So I will just show you, since I will stop at 11:30. Okay, I will show you these three examples. Just before we go a bit to the things we can do with this cell-free approach, I will just show a first part about synthetic cells. So synthetic cells, maybe some of you, I heard some people working with membranes, so some of you maybe work a bit, some of you maybe don't know. So usually in this field of synthetic cell, usually what people do is that the cell is very complex. I showed you yesterday that you have this complex architecture. So maybe you can reconstruct something that is control, and then you can study. So if you do this kind of, you look at what is the essential break. Of course, a membrane is much more complicated than just lipids. You have 50% of lipids, 50% of other things, many proteins. But you can take this elemental break phospholipid, which is a bit like a surfactant you saw yesterday, except that they are this cylindrical shape. And then you can do the opposite behavior. You start from this elemental break and swell it, and then build this kind of bag, and with a phospholipid bilayer, and then you get something that is a kind of cell model. And this is called a giant liposome. And this can be typically between 1 to 100 micrometers. So I have something that is very far from a natural cell, but that can be a very simple model because you have a membrane, and you have an inner compartment, and you have an outer compartment. So actually, we have developed these things. We have developed methods to control their size, to control their composition. We can even engineer some phase separation, so you can make some kind of membranes that look closer to actual membrane. And I just show you, this is quite interesting. It's a recent work where we confine this liposomes using macrofluidics. So, for example, we force these cells to be elongated. Because most of people, they do artificial cells, but they do just spheres. And you agree that this is very far from reality. All objects are not isotropic, they are anisotropic. So we had this idea which we can just use microfluidics to elongate this synthetic cell so we can control the shape. I know what is very interesting is that if you take a mixture of lipid to have phase separation, liquid-liquid phase separation, you see that instead of being random, this phase separation starts to have specific distribution. And you see is that in this you see that here you have two types of phases. You have in red a phase that is liquid disordered, very fluid. And in blue, a phase that is liquid ordered, so that is a phase that is more organized, like a liquid crystal. And you see that if I play it again, at the beginning, I have a mixture of these two phases, and then when I deform my vesicles, this phase in blue will go to the extremity, and the phase in red will become in the middle. Okay, so I can create some poles like that. And what is super interesting is that it's not controlled by the chemistry, because if I if I revert the composition, if now I just take the same lipid, but I have a majority of the red phase, the liquid one, then it will go at the extremity. And then in the middle, you have this blue liquid order phase. So in fact, it's very interesting because you can create poles in your synthetic liposomes like what cells do. And it's actually not controlled by the chemistry, but just controlled by, in this case, the line energy. So the system tried to minimize, in surface tension, we are in 3D, we have a surface that is in 2D, and then the system tried to minimize the surface. That's why droplets have a round shape. But because now we have a membrane, so this phase separation is in 2D, so what counts is not a surface tension, but it's a line tension. So it's the energy cost by creating this frontier. This is happening in a channel, so this liquid phase separation, and with that, then you can have some synthetic cells where you can control by this physical process how to put this liquid domain. And then if you start to make complex shape, you can control the positioning of these domains, making different kinds. You can make synthetic sets with 3 poles, 4 poles, and so on and so forth. So just that it's possible to control these membranes, but it's still very passive in terms of functionality. And then what is very nice is that with this cell-free expression, you can put your DNA inside with your code, and you can start to produce protein inside these bags, and you start to have something that looks closer than what the cells did. And I show you this is a very old paper, but it was the first time that proteins were expressed from purified components, or from what I showed, the cell-free expression. So it was the first time that we can have a fully synthetic system, So we have a synthetic membrane that we build. We have the synthetic DNA that we put inside. We have the C3 measure is fully synthetic. And all these things are able to produce protein. So it's very simple. It's fluorescent protein, nothing fancy compared to your IDs. But it was to show that we can do something that starts to be a little bit elaborate in terms of mimicking cells. And then, of course, you can go to a bit more interesting functionality. Here we do a membrane protein that is accumulating to these liposomes. And this membrane protein is interesting because it's kind of, it's in a membrane. And this protein is a pore. It's an active pore, so it's in a closed state. And it's a bit like a mouse. It can open and close. And what is interesting is that this pore is closed, but when two cells come together and when these two pores come in contact, they open together and they make a channel. And this is used by cells to exchange medium. So it's a kind of something like that. It's called the gap junction protein. So you have your protein is here. It's closed. This is a cell. And this is off. This is closed. And when you have your two cells together, they kiss. It's like a kiss. And when they kiss like that, when they kiss like that, they will open a channel and then they can exchange medium. So it's a very interesting system for cells to communicate. Usually cells, when they want to communicate, they sell some molecule outside, so they have to do exocytosis, and then this molecule diffuse a lot, and another cell will capture it. And here they can do this to directly communicate, for example, hydrophilic molecules without needing to go outside. So we can make this kind of functionality. And okay, so then there is just imagination of the limit. And now I would like to show more functionality. So when we discussed with you, you had a lot of examples of enzymes. We also did enzymes, I didn't show in this talk, but we did enzymes, for example, that can convert molecular prodrugs, so non-toxic drug, to active cancer drug, okay? So it's quite interesting because when you do cancer treatment, you need very aggressive drugs that can kill the cells. And the problem is that when you do this, it kills other cells and you have all these side effects of cancer treatment. So what is nice is that if you take a prodrug, so it's a drug that is inactive, and then you need an enzyme usually to cut this drug, and then this drug becomes active. So if your enzyme is close to your tumor, then you can kill the tumor and not too many other cells. So what we did is that we thought a bit like what you said there was this idea of making therapeutic. Here we say, okay, this enzyme is very useful, so we just take its sequence, as I said, we go for the DNA sequence, and then we take this DNA, and we express it, we put the prodrug, and we can release the active drug. And if, in our case, we did photo control of gene expression, so with light, you can trigger the expression, and then you can trigger the release of these active drugs where you want. So just to give an example of what you can do, you're just limited by imagination in this approach of programming from DNA any protein you want. So I will show you one example because it's recent and I think it's quite interesting in terms of impact. And then at the end, I will finish with really soft matter things. So this is in the context of antibody discovery. So you know, I think nobody suggests antibody, but they are very important proteins. They are the proteins that recognize. So they are the basis of all immune systems, are antibodies. So it's the basis of all how life is working, how life can recognize itself or other cells so that it can decide what is myself, what is not myself, and then make systems that can have their own individuality and fight against aggressive systems. This is the base of immunology. And everything is based on recognition and is based on antibody. Okay, so antibody are really incredible molecule because they can recognize very specifically some target. So it's very important for the biology, but it's also extremely important for application because if you are able to have an antibody that can recognize specifically something, then you can really start to think of making very interesting application. Antibodies are used for all your diagnostics, all the tests that we know, COVID tests and so on, they are based on antibody recognition. And now the huge interest in therapy is to use immunomedicine, so it's to use antibody to treat people, and especially cancer. So it's a huge field, and in a pharmaceutical company, currently there is, I think, more than half of the investment is for antibody discovery. So pharmaceutical company, they don't want to synthesize organic molecule anymore. They want to use proteins, and they want to use mainly antibody. Okay, so this is the new frontiers of pharmaceutical research now is antibody. So they want antibody that can recognize either cells or either disease, and then they can use this antibody to block the disease or to recognize it or to train cells to become aggressive. So this is probably the future of cancer research, is that instead of getting an external component like a drug to kill the cancer cells, the idea is that you train your immune system. So you take a patient and you take the patient immune system, so its own cells, and you will train this immune system to recognize the cancer cell, so that the patient will use its own immune cells to kill its own cancer. So this is really what is the future probably of, together with vaccine, is probably the future of cancer therapeutic, is immunotherapy using so-called CAR T cells. Probably you have heard about it. But basically, you take the cells of the patient and you train the cells. And all is based on antibody-antigen recognition. So there is a very huge importance in terms of biotechnology and medicine in antibody discovery. And this is how it is made now. How you discover an antibody for therapy. I don't know if you know, I didn't know before I started to collaborate with a Sanofi company, it's a big pharma company. So typically, it's a awful process. It's a awful process. You take an animal and you put a disease in this animal, and you let this animal with a disease, and then you make a soup of this animal, and then you will try to fish the antibody. So it's a horrible process. It takes a lot of time, it takes a lot of money. It's typically 6 months of intense research, millions of dollars of investment, robots, large-scale approaches, a lot of people. It's very long, it's very costly, and it's mainly animal-based. So I don't care so much by the fact that it's long and costly, because pharmaceutical company can manage this problem. But I have a problem that is also animal-based. So it's very, very middle-age approach. You know, you take something alive and you make it, you kill it, and you try to see. So could we change this way of doing antibody discovery? Right? Could we make it in a way that could be a bit smarter and maybe more efficient? So the idea is very simple, is that instead of putting a diseased animal, make a soup, fish the antibody, look at the sequence of the DNA, make all this billions-dollar process until you find your antibody. Maybe we can do something very simple. Maybe we can just take the DNA and just express the antibody from it and test if this antibody is efficient. And if it's efficient, I keep it. And if it's not efficient, I trash. And to make it efficient, I don't need kilos, I don't need a horse. I can just do it in a small drop. So, you take a picoliter drop, just a few micrometer. So you have 10 minus 12 liter, and then you can test your antibody function. So you see, you can reduce a lot. And of course, to go from DNA to this, we can just do cell-free. So it's very simple in terms of concept. You just take a drop. This drop is a few picoliter, just a tiny drop of a few micrometer. Inside, you put your DNA. This DNA, you put the sequence of an antibody. It's called VHH. It's VHH is antibody you find in camels. You know camels and llama. They are very interesting animals. Llama, you don't have llama in India. You have many other animals. I don't know, maybe you have camels in India? Yeah, you have camels, so great. Okay. So camel, it's, llama are the same family of camels. They are called camelids. And they have this very interesting antibody is that instead of having this very complicated antibody as many of us have, they have a very simple antibody. Very simple. And our antibody are typically 100 megadalton size, and these guys are very, very small antibody, very short. It's called a nanobody. And this is super cool because they're very small, so they're very easy to synthesize, and they are as efficient as our antibody. You can find similar in shark. Okay, so shark, llama, camelids, they have this super smart antibody. They are very small. So we took this because they are easy to synthesize. So you put the sequence, you put in a drop, and the idea you just do, you put your cell-free expression medium. It's very simple. And then your antibody will be expressed inside. These are some magnetic beads. And then when the antibody will be expressed, it will attach to these beads. And then its target, its antigen, is, for instance, fluorescent. So what will change is that you have overall fluorescence, you express the antibody, and if your antibody is active, the fluorescence will be captured. So then here you have homogeneous fluorescence, and here the fluorescence is concentrated. Okay, so we just do that, and then you can measure the activity of your antibody directly inside a single drop, a single picoliter drop. And it actually works very well. So if you take, we start by an antibody that we know it works. So we put the DNA inside, we express it, and you see that you have fluorescence that is accumulating on this magnetic beads assembly. So it means that the antibody is capturing the antigen. Okay, so and you can do inside each drop, you can make some kind of a binding assay, so you can measure the binding affinity inside each drop. Okay, and with this, we can, it takes 3 hours to do this. Basically, the only time we need is to do the expression. And in the 3 hours, we can measure the binding affinity of your in situ synthesized DNA. So this is done in 3 hours, and it's done with 1 million smaller amount of materials than regular assay. So here we have already a 1 million gain in terms of efficiency. And another interesting aspect is that here I have drops, and in this case I just put the same DNA in all my drops. But now, when I synthesize my DNA, I can, on purpose, just start from a library. Okay, so I can put a lot of different DNA, and I can look at the formation for each single drop. Okay, so now I can go, I can send my drop, and I can measure the efficiency for each DNA. So we tried, for example, for a mock library, so it's a fake library to test the efficiency of our method. But for instance, we put one positive efficient sequence in 1 million of sequences that express an antibody that is less efficient, and we can recover it with something like 80% of accuracy with just 2 rounds. So we just do this twice, and we are able to fish this interesting sequence. So now we have a system that is fully synthetic. There is no cells, there is no animal, there is no cell culture and everything. It's fully animal-free, and we just take from synthetic DNA. It's highly programmable. You can take any sequence you want, so you can screen any types of antibody you want. And not only you can synthesize a given antibody, but then you can make mutation. So you can explore for new antibody or better activity. You can start to imagine maybe there is a function that doesn't exist in nature, but I want an antibody that recognizes this or that, and then you can screen a lot of sequence until you find the antibody you can do. And just to give you numbers, now we are applying to actual library, so to find new antibody for medical application, for therapeutical targets. So now with our assay, in 2 weeks, we can screen actual library. So now we are looking for unknown sequences. So in 2 weeks, we can, after 2 weeks, extract a positive sequence that will express an efficient antibody against a therapeutic target. And 2 weeks is great, and this is just done in a chip that we do in the lab. So it just takes one bench size, 2 weeks, no animals, and you can screen libraries, which is against 6 months of massive industrial scale conventional approaches. So now I would like to go into implementing this idea of synthetic biology, self-expression in very soft matter cases. So as I said at the beginning, the idea is that instead of doing formulation, we would like, so instead of trying to put everything I need until I find my target properties, I want to put DNA inside that will program everything. So we'll try to do, I will show you 2 examples of this idea of putting a genome in a chocolate mousse. The target of one target we wanted to do is to do surface functionalization. You know, surface chemistry is very complicated, and if you ask people to do even more complicated, is liquid surface functionalization. So if you ask somebody, you take a liquid surface and you ask, you want any kind of chemical function, this is very hard, you know. It's already hard on a solid surface, so imagine on a liquid surface. Oil is very inert, water is very inert. How do you surface functionalization? It's very hard to do with conventional chemistry. So we said, okay, let's find an interesting protein that could do that. So you just look at literature. I found this protein. It's interesting. It's a self-assembling protein, like a surfactant, right? It's from bacteria that makes this biofilm. So it's self-assembling, and it's very interesting. It coats surface, this protein. So this could be very nice to do surface functionalization. Maybe if we express this protein, we can start to functionalize liquids. So it's very simple. Again, we just take the sequence of this protein. It's published. We put in the DNA. We just take a regular oil drop, and we will express this protein. And we hope this protein, because it can coat surface, that it will naturally coat this oil surface spontaneously. And then because it's fully programmable, you just put any DNA you want here, and then you can express any interesting protein, any protein of interest of your choice. So this is the strategy. We can start with simple protein, fluorescent protein. You see my droplets, regular emulsion. If I express my fluorescent protein, I have fluorescence, but my droplets are not functionalized. But if now I add this bacterial biofilm protein here just before, then it's expressed, it will transport the fluorescent protein on the surface, and now we know that you see my proteins, they become fluorescent. Okay, so you can program a protein that will bring all this fluorescent protein on the surface, and you can rinse, and you have this nice fluorescence coating. And no, it's nice because you have programmability, so you can start to implement programs. So for example, you can add a small sequence in your protein that will be specifically cut by some protein. And with that, you can have a function. You add these proteins, and this function will be released. Okay? We don't care so much about all these properties. What I think is interesting is the approach. Okay? You can put all this information in your DNA. You can also do this antibody. So you can now say, okay, I want droplets that can recognize a target. Easy. I just add, I put a sequence that can express an antibody, and now I have droplets that have antibody on their surface. And you see these droplets, they can capture the antigen. Here it's a GFP protein. And if you put another protein, they will not capture it. So you can make a functional antibody on that. So then you can start to do some interesting materials, like here, we take these droplets, functionalize with an antibody, and then this antibody can recognize the cells. So now you have droplets that can recognize cells like this, and you see that if I flush here, the antibody is not expressed, so the droplets are escaping, but if they are expressing the antibodies, they remain attached to the cell. So you can start to build materials that have some kind of interesting properties. So here are, for example, drops that can recognize some protein, some cells that express some specific protein on their surface. So you can start to build some interesting materials. So I just give here examples that you can functionalize with fluorescence, with protein release, or with recognition. And to finish, I want to finish with this idea of making a dosa that can move, or making a synthetic object that can move. And I want to program it from DNA. I showed you yesterday that interfacial dynamics can lead to a lot of interesting dynamic phenomena. So you can have droplets that move, you can manipulate with magnets, you can manipulate with light, and so on. So we know how to make things move by using soft matter approach, right? And you have done some laboratory hands-on experiments yesterday. You can do mahogany, you can do all these kind of things. So could we do that from DNA? Nobody suggested this when I asked you, can we make something move from DNA? Okay, what kind of protein I could do if I want my objects to move? So what kind of protein I could have, I could synthesize to make this? Here, again, yesterday I say all these things are based on surface tension, surface tension gradient. So can we have a protein that could control this? And then I could program this into a DNA so that I could have a DNA that expresses this so that I can have all this property. Could we get a protein doing that? Maybe it exists. So you just look in literature and you look for protein that could have this kind of, could control surface tension. So you don't look for a protein that makes something move because you are smart and you say, I don't care about the biology, I care about the property. So I'm looking for a protein that controls surface tension. And you just look, and you can find in some papers some proteins from mushroom. So I found this some old paper done in some microbiology people working with mushrooms, you know, so something very far from our field. And they describe this protein called hydrophobin. And you see the title, the protein amphiphiles of filamentous fungi. You have this super interesting word here, amphiphile. So you have these mushrooms that produce surfactant. And these surfactants, of course, are proteins because they are alive. And I found it super interesting because this protein, if it's amphiphile, it's kind of surfactant. So now I could program the synthesis of surfactant from DNA using biological just cell-free expression. So maybe we could do genetically encoded surface tension or genetically encoded liquid transport. Maybe we could put a DNA that will control surface tension. Nobody has done that. So that could be very interesting. So we did that. So we put the sequence of this strange mushroom protein or DNA, you do the cell-free expression, and you measure surface tension. Simple experiment. So these are the results. If you don't put DNA, you have this kind of surface tension. If you express a fluorescent protein, you have nothing special. Fluorescent protein are not surfactant. But then, if I express my strange mushroom protein, look, I decrease surface tension. I decrease surface tension by gene expression, and nothing has done that before. And you see that we can do that with what we know about knowledge. So, okay, I was really super happy. I put my DNA and I decrease surface tension, and then you can say, okay, if I take soap and I put in water, I have also decreased the surface tension, right? Okay, so it's kind of pressure of a scientist in a strange lab somewhere, but okay, you can do this easily, right, if you want. But maybe with this we can do something that is difficult to do with soap, because now I express this protein. What is interesting is that now I can program the expression. So if I control the dynamics of expression, I can control the dynamics of this surfactant directly from my system. So for example, here you see if I have continuous expression, surface tension decrease and stabilize. But then if I turn on expression, surface tension decrease, I turn it off, it becomes stable, I turn it on, and then surface tension decrease again. And then you can start to make some complex shape. So you can start to program dynamically the temporal evolution of surface tension directly from your system. So you start to have very elaborate properties by using this programmability approach. So now let's finish with the moving DOSA. We do with more simple system, just water. Okay, so the experiments are following. I have water in a capillary, okay, and my idea is that if my system is static, when I will put my DNA, I will express this protein, surface tension decrease, and I want to see this guy move. So here is the experiment. You see, without DNA, my meniscus is almost static, and if I have DNA, I have protein expression, and my droplet is moving. So it's a very simple experiment. It's a droplet in a capillary. Yes, we are very far from complex, massive transport, but it's the very first time that we can program something like a motion from a DNA, okay, from a programming. So I can put in my DNA the information that my droplet should move, and then my droplet is actually moving. So the first time that we can combine these two aspects. And because it's DNA, then I can put in my DNA what you say, like DNA that express an enzyme, DNA that express an antibody, DNA that express a molecule that degrade plastics, DNA that express whatever you can imagine of, and you can combine it with motion now. So you can really think to build maybe some interesting new materials that are actually smart because you can put information inside. And again, if you have dynamic control of expression on and off, you see that if I switch off expression, the system stops. If I switch on, it starts. And again, and so on and so forth. So I think I show you that it's possible to put in some synthetic system a DNA that will program a lot of properties. So it can be just functions, fluorescence, enzymatic activity, protein release, antibody recognition, or some quite interesting properties. Surface tension, liquid transport, even viscoelasticity that I didn't show it, but we can also control the viscoelasticity by this approach. I would like to thank people all the work I showed today. And I think we had quite a lot of discussion during this lecture, so maybe we can just stop for now, but I'm happy to discuss at any time during the day.